You've seen a Khan Academy video giving you an overview of the process of transcription. I want to dig a little bit deeper into the chemistry that's going on behind that process and then separate what's going on in transcription in both prokaryotes and eukaryotic cells, starting, of course, with the simpler prokaryotic organisms. So to begin with the chemistry, we have to talk about the organization of atoms within the ribonucleotides that are going to be polymerized during that process of transcription. So we've got a little diagram of a ribonucleotide here with the pentose sugar, ribose, demonstrating that the pentose sugar, meaning the five-sided ring, has an oxygen in one position, and adjacent to that is a carbon atom that's in the number 1 or the 1' position. To that 1' carbon, a nitrogen-containing base is going to be chemically bonded. To the carbon in the 2' position, there is a hydroxyl or an OH group. That is also true in the 3' position. So the carbon in the 3' position is going to be bonded to a hydroxyl group. The 4' carbon will be chemically bonded to a 5' carbon that is outside of that pentose ring. And a variable number of phosphate groups will be bonded to that 5' carbon through an oxygen. Note that the numbers that we are giving the carbons in this pentose sugar have a prime after it, so 1', 2', 3', 4', 5', and that is because the carbons or the atoms that are in the nitrogen-containing bases are also numbered 1 through potentially 9. Without that prime designation. So, for example, 1, 2, 3, 4, 5, 6, 7, 8, 9 in a purine nitrogen-containing base. One more thing, you'll note that a nucleoside is the pentose sugar with a nitrogen-containing base. A nucleotide is that pentose sugar plus a base plus a variable number of phosphates. If there's 1 phosphate, it's a monophosphate. 2 phosphates, it is a diphosphate. 3, it's a triphosphate. And the phosphates are also designated with Greek letters, the alpha phosphate being the closest to the pentose sugar, the beta being the one in the middle, and a gamma phosphate being the one furthest from the pentose sugar in a nucleotide triphosphate. When we polymerize mononucleotides into what's shown here, a dinucleotide chain, here's what's going on. We have 2 nucleotide triphosphates here, ribonucleotide triphosphates, and we know that because there is an OH or hydroxyl group attached to that number 2' carbon. The bond between the alpha phosphate and the other phosphate groups is going to attack the oxygen that is part of that hydroxyl group attached to the 3' carbon. That will form a phosphodiester bond. So here is the alpha phosphate that is attached to an oxygen attached to the 3' carbon. That phosphate is also attached to the oxygen that is attached to the 5' carbon of the other or the second nucleotide. So again, this dinucleotide, air quotes, chain will have polarity in that there will be a 5' end of the chain that will be associated with 3 phosphate groups bonded to that 5' carbon, and there will be a 3' end of the chain which will have a hydroxyl group associated with the carbon that's at the 3' position. Polymerization will always occur dependent upon a DNA template. So here we have the template strand at the bottom, and of course we are going to use Watson-Crick, typical ATCG base pairing, in order to know which ribonucleotide triphosphate we are going to bond in that polymerized chain. Here we have a guanine ribonucleotide triphosphate that would not bond to this adenosine, so it's going to be kicked out, so to speak. But if this were a U nucleotide, it would bond to the adenosine, and the alpha phosphate would react with the hydroxyl group that's attached to this 3' carbon, and that creates a polynucleotide chain. We are going to polymerize nucleotides, always adding on to the 3' end of a growing chain. Polymerizing those nucleotides, we say, in a 5' to 3' direction. Again, we're adding onto the 3' end always, and we are reading the DNA template in the anti-parallel or opposite direction, reading 3' to 5'. In the end, we have a ribonucleotide chain, or an RNA nucleic acid, that has a series of ribonucleotides bonded by these phosphodiester bonds. Again, 5' end and a 3' end, demonstrating polarity in that ribonucleotide chain. The RNAs that can be produced by the process of transcription come in a number of flavors. Of course, in high school, we've heard about mRNAs, messenger RNAs, and those are responsible for coding for proteins. Ribosomal RNAs that form the core of ribosomes, so those are rRNAs. We also have tRNAs, so these are transfer RNAs that are used in the process of translation or protein synthesis. But we also have a couple of other flavors, microRNAs, miRNAs, that are responsible for regulating, we'll say, the process of translation, so regulation of gene expression. And we also have hnRNAs, or heteronuclear RNAs, that are involved in nuclear processes, about which we'll hear some of in probably tomorrow's preparation. Again, a reminder of what the Khan Academy video said is that the process of transcription has 3 steps. The first of those steps is initiation, transcription initiation. Then we have the elongation step, which is simply that polymerization of ribonucleotides using the DNA template, reading that template in a 3' to 5' direction to polymerize ribonucleotides in a 5' to 3' direction. And then we have the process of termination. The Khan Academy video was a little bit confusing about that termination, so I want to talk a little bit more about that when we get there in a few slides.